Hi guys. Good morning. It is week 3 of the semester, and you people have a holiday on, well, we all together, all of us have a holiday on Friday. So, in order for us to continue on in our semester, I wanted to do a quick recording of a portion of the lecture that you guys are going to miss. And the topic of the lecture this week is biomolecules. So I want to spend just a little bit of time kind of introducing these organic molecules to you. We're going to revisit them quite a bit during the semester, but this purpose of this was just to kind of get acquainted. So the 4 biomolecules that I want to discuss briefly today are the carbohydrates, the lipids, the proteins, and nucleic acids. So these are molecules that are going to all have carbon, of course, because they're organic molecules, and they are super big. These are really large molecules. So last week in class, we kind of introduced the concept of how we start to build things out of different elements. So we learned about subatomic particles, and we learned about how the kind of the configuration of the arrangement, I guess I could say, of these subatomic particles is what allows molecules to interact with one another. And of the 3 subatomic particles, if you remember, the one that we said was the most important in terms of the relationship of one element to another and how likely it would be to bond with another element are the electrons. So the electrons are found, if you remember, in the outer shell. And and again, if you recall, the goal of the electrons was to, or is to, always try to fill those outer shells. So if an element has an outer shell that's almost full, it's going to be on the search for another electron or another couple of electrons. Conversely, if an element has just maybe 1 or 2 electrons hanging out in its outer shell, and it's not the first shell, of course, which only holds 2, then that particular element is likely to have its electrons stolen. So when we talk about sharing or stealing electrons or transferring electrons from one element to another, the relationship that results as a result of that behavior is a bond. And we talked about a couple different bonds last week, one being hydrogen bonding, which are really weak bonds but give a lot of structure, and we'll talk a little more about that today, and also covalent bonds and ionic bonds. Ionic bonds, if you recall, result from the complete transfer of an electron or the stealing of an electron, and then the relationship in between those 2 elements then would be an ionic one, an ionic bond. And if the electron or electrons are shared between the 2 elements, then we call that a covalent bond. So when we start linking these things together, we make big molecules, right? And so that's essentially what we're doing here, talking about today, is these big macromolecules. So they are the carbohydrates, the lipids, the proteins, and the nucleic acids. So essentially what we see oftentimes, actually with 3 of the 4 of those particular biomolecules, we see a repeating unit. So if a unit in a molecule is repeating, like for example, if you have repeating units of sugar molecules like glucose, if you have just the single unit would be referred to as a monomer. So if that green dot here is indicative of a monomer, if we start linking them together, we call them polymers. So a polymer is just a repeating unit of a single monomer. So like if that was a big sugar molecule down there, we would call that a polymer, right? So in the lab, I said to some of you who are in my lab, if you have one sugar, we call it a monosaccharide. If you have we call it a disaccharide. And if we had lots, 3 or more, from 3 to however many hundreds, we would call that a polysaccharide. So a polymer is just a kind of a generic term for a large molecule that's made up of a repeating unit. All right, and so the molecules we're going to talk about today are organic molecules, and again, if all organic molecules contain generally a carbon and a hydrogen bond, but absolutely contain carbon. There are no macromolecules or biological molecules that do not contain carbon. Okay, so let's look at our 4 classes. And again, we're going to talk about these throughout the semester, so today's job is just to introduce them. And so here's a kind of a quick introduction. You can see the 4 boxes. Each of the 4 boxes has a description of one of the 4 macromolecules. Up on the top left, we have the carbohydrates. Carbohydrates are generally made up of repeating units of sugar. So that picture there is a starch molecule, and starch is a very large repeating unit of glucose. So the monomer in this particular example is the glucose, and you can see it's in that little box there in the middle. In terms of a summary, the function of carbohydrates, they're a quick energy source, so we can make sugar out of them. We're going to spend a lot of time talking about that in the next few weeks. And the other thing that carbohydrates are is they're pretty structural. We see carbohydrates all over the place. We see them in hanging off our cell membranes, which we're going to talk about later on this week as well. But they're really important energy molecules. Moving to the right, you see the proteins. Proteins are really structural. They tend to be pretty globular, like that one. You see lots of things kind of all stuck together. Of all of these macromolecules, probably the most abundant are proteins, and they have a whole bunch of functions. So, for example, all of our enzymes in our body, enzymes are proteins that are required to assist chemical reactions progression. So that means that, well, I could say that all chemical reactions in the body, in the human body, would not exist on their own. They need an enzyme to catalyze that reaction. And a catalyst means it's simply something that kind of gets something started, right? So enzymes are catalysts, and enzymes are proteins. So proteins serve as catalysts. They're also really structural, so molecules that are big and like kind of elaborate in terms of their form, those generally have a protein component. Our immune system relies on proteins for defense. We make things called immunoglobulins or antibodies, and those are proteins as well. We'll learn a little bit about those later on in the semester. Oftentimes, proteins are used to transport substances from one place to another. Like, for example, in our blood, we have plasma proteins, and the plasma protein's job is to pick something up that wouldn't transport through the blood on its own. So traditionally maybe something that's either charged or not water soluble, and carry it around. So they're big transport molecules. And also there are a lot of our hormones, which we use for distance communication. Those are also made out of proteins. So there's a lot of roles for proteins in the body, and we're going to talk about them a lot over the coming the next like 14 weeks or so. I'm going to come down next to the nucleic acids. Nucleic acids there's 2 that we're going to talk about. One is deoxyribose nucleic acid, abbreviated as DNA, and the other is ribose nucleic acid, abbreviated as RNA, and they're really important because they are where all of our genetic material is contained. So we are going to learn a lot about DNA and RNA and the roles of DNA and RNA in terms of cellular replication and function. Of the three that we've talked about so far, the carbohydrates, the proteins, and the nucleic acids, those are all made up of repeating subunits. The carbohydrates are made up of repeating subunits of glucose or another sugar. Proteins are made up of repeating subunits of things called amino acids. We'll see that in a little while. Amino acids are the structural, the functional unit of the protein. Nucleic acids are also made up of repeating units of nucleotides. So these are just large molecules that are basically the same thing strung together. The fats, or the lipids, to the left here, our very last category, these ones are different. These are not made up of repeating subunits. There's very different classes of lipids, and we'll talk about a few of them today. They're also a really good, actually the preferred energy source in the body are lipids. We'll learn more about that. We can make a huge amount of energy from them. The process is a little bit different depending on the type of fat or lipid. They also serve as insulation for us, keeping our inner essential body parts warm. Fats are really important in terms of hormone regulation. We have cholesterol, this is what we're looking at right here. This is a molecule of cholesterol. That's an example of a lipid. The cholesterol molecules are our steroid hormones all have this cholesterol backbone as well. And again, hormones are communicating molecules. And also cholesterol is embedded in our cell membranes, and it makes your cell membranes fluid. It makes it so they're not so rigid. And a fluid membrane on a cell is a much more functional setup than a rigid membrane. So we'll learn more about those. So today, really, what we're kind of trying to get a sense of are these four big macromolecule or biomolecule classifications. So one more time, they're carbohydrates, proteins, lipids, and nucleic acids. So I want to just kind of point out some significant features of the individual macromolecules. Of course, it's not going to be an exhaustive conversation because this is, we could almost spend a whole semester on each kind of macromolecule. But I want to just orient you, get you familiar with them, so when we see them again, you'll have some sort of place to work from in terms of what these things are all about. All right, starting with the carbohydrates. So again, I said earlier, this is a quick energy source, right? So most of us know that if you're dragging and you eat a little sugar, you kind of get a little perk. And that's because that sugar gets converted very quickly into our energy molecule in the body called ATP. Again, carbohydrates are made up of the repeating units of sugars, so that's why they're called, at the most basic level, it would be called a monosaccharide. So an example of glucose would be a monosaccharide. If you link two together, we're going to call those disaccharides. And if we link more than two, we would call them either an oligosaccharide or, as it gets pretty complex, a polysaccharide. So again, what we're going to appreciate from these molecules is they're a very quick energy source. So when we learn about cellular respiration, probably a week after right after your first exam, we're going to learn how to burn sugar for fuel, right? And it is a pretty elaborate process, but it's pretty interesting. So here we have three examples of monosaccharides, right? So here is our glucose. You can see it's a carbon-hydrogen bond right there, always. More than one in most cases, more than one carbon-hydrogen bond, as you can see. So this right here, C6H12, this is the molecular formula for glucose, which means that it has 6 carbons, 12 hydrogens, and 6 oxygens. We have fructose here, which is also a monosaccharide. This is the sugar that we find in fruit. It's a tiny little bit different in terms of structure, but it's very similar. You can see the formula is the same. And just the configuration is different. And then you have galactose, which is our other monomer of sugar, right? So if you were to put two of these together, glucose and fructose, right, that would be the reaction that allows for that to happen. The linking of these two things together is called dehydration synthesis, because basically what we do is we take out a molecule of water and then we add these guys together. So essentially what you're doing right here is you're taking out 2 hydrogens, this one and this oxygen, so that's water, right? H2O. We remove 1 molecule of water, and you can see it leaving here, and then a bond forms in between. That's called dehydration synthesis, when a bond forms when you remove a molecule of water. And so when you join glucose and fructose together, we end up with something called sucrose, which is table sugar, right? That white sugar that you put in stuff if you're baking or putting in your coffee or whatnot. If we wanted to break this sugar apart into its 2 monomers, that process is called hydrolysis, and you can see it right here. So what you do there is you add a molecule of water to sugar. You throw some water in here. This bond would break. One of the hydrogens would link here. Another OH hydroxide would link here, and we'd have 2 separate monomers, glucose and fructose. So those 2 terms are terms we use for chemical reactions. A chemical reaction is anytime 2 things interact together and either something new is formed or a bond is broken. Either one of those would be a chemical reaction. Generally, an enzyme is required for that to happen. And the linking, again, of 2 different molecules is called dehydration. Synthesis is to make, and lysis is to cut. So dehydration reactions or dehydration synthesis makes new molecules, larger ones, in this case a disaccharide. Hydrolysis breaks things apart. All right, so here's some pictures of some really big sugar molecules. that we may talk about during the semester. Starch is what we find in potatoes. It's just repeating units of glucose. We talked about starch in the lab last week. Glycogen is how we, as humans, store our sugar, and it's again just repeating units of glucose. We call it glycogen. That's found in our skeletal muscles in large quantities and also in our liver. Cellulose is what plants use to link their sugar together. Cellulose is undigestible by humans. It's like fiber. And chitin, you can see that in that tick, my least favorite critter on the planet. Well, probably, or at least way up there. And again, chitin is just how the tick stores up its energy, repeating units of sugar, right? So starch and glycogen are like our food warehouses, whereas the cellulose and the chitin are more structural in the plants and also in the tick. But again, it's all simple sugars linked together, so it's a carbohydrate, repeating units of sugars. All right, so lipids, these are the ones that are not repeating units. They're going to have a lot of carbon, a lot of hydrogen, and a lot of oxygen. Because they're fat, they're not going to dissolve in water. And anything that's not soluble in water, we call that hydrophobic or water-fearing. So you're going to want to remember that term. Hydrophobic means water-fearing. So there's basically 3 types of lipids that we're going to look at. One are the triglycerides, and that's just a fancy word for the fats and the oils. Then we have the sterols. That's what steroid hormones are, and cholesterol is a sterol. And then we have the waxes. So these are all a little bit different structurally. They are going to all have carbon, hydrogen, and oxygen, and a large quantity of hydrogen, and they're all going to be hydrophobic. But they're not all exactly the same in terms of repeating units. So fats can be kind of confusing. Really what it all more or less comes down to are the bonds, right? So it all comes down to what kind of bonds are holding these things together. So you may have heard of some of these terms before: saturated, unsaturated, and trans fats. So a saturated fat, and again, really it all kind of comes down to bonds. Unsaturated fats tend to be liquid at room temperature. So like an example of an unsaturated fat would be olive oils. A lot of the vegetable oils are examples of unsaturated fats. And it sort of has to do with the way the temperature, the consistency at room temperature, and also how many double bonds they have. So an unsaturated fat is going to be liquid at room temperature. A saturated fat tends to be solid at room temperature. So an example of a saturated fat would be like butter is a saturated fat. It gets a little bit kind of more confusing. In our world, fat is it's kind of a confusing topic in terms of healthy fat versus non-healthy fat. And traditionally we think unsaturated fats are healthy and saturated fats are not healthy, but that isn't totally true. The evidence is showing that saturated fats actually can be very healthy as long as it's a good quality fat. So for example, like butter is gotten kind of a bad rap, and we think of butter as being bad, but it's not necessarily bad. It's the quality of the butter and how much and the quantity that's being consumed, if you will, right? Fats are a really good source of fuel. That's what we have to remember. Fats are a great fuel source, and as long as it's a good quality fat, it's not rancid or been processed too much. Saturated fat can be quite good because it's going to be a good fuel source, but we have to sort of think about it in terms of moderation and quality again. Trans fats are the ones we want to kind of stay away from. I'll show you a picture of those in a minute. So remember, we had 3 categories. One are the triglycerides, which are the fats and the oils. So these are the ones that I was just talking about. So at the top here, so this is a triglyceride. So it's got what we call a glycerol head. So this up here is a glycerol, which is an alcohol, and it's bonded to 3 fatty acid tails, right? So you can see these things. So that's why it's called a triglyceride. It's got 3 fatty acid tails bound to this head up here. So look at all the hydrogens and carbons. Lots of hydrogen-carbon linkages here. All right, so again, saturated fats. So saturated fats have all straight fatty acid chains. So see how this is straight? That's called a saturated fat. Basically, all the carbons are saturated with hydrogens is what they're saying. An unsaturated fat is going to be one that has at least one double bond. So we're looking here. So we're looking here at this double bond between these two carbons. So when you form a double bond, right? So now this carbon is not saturated with a hydrogen. Instead, it's going to form this double covalent bond with this carbon. So when that happens, what it does is it causes the tail of the fat to kink. So you can see it's got one kinky chain here to be an unsaturated fat. It has to, pardon me, have at least one. This one has two. It has to have at least one kinky chain to be an unsaturated fat. Again, our unsaturated fats are our plant-derived fats traditionally, and they usually are liquid at room temperature. Our saturated fats are generally our animal fats, and those are solid at room temperature. Okay? Again, we're not saying one's better than the other. It's just structural at this point. Okay, so I have a couple other pictures in here for you if you're interested. So the unsaturated fats, those are the ones we traditionally think of as the good fats, right? Those are the ones we find in nuts. We find them in avocados. Of avocados, you might be thinking, well, an avocado isn't liquid at room temperature. Avocado oil, however, is, if you just take the fat out, the avocado oil is liquid at room temperature. The saturated fats, these are generally our animal products, butter, fat, meat, the fat you see in meat, rendered fat, those kinds of things are unsaturated fats. The unsaturated fats, I should say, are really stable. One of the things that seems to be bad about fats is the instability of them. And so in terms of how, just for some practical knowledge, one thing about the saturated fats, is because they're very stable, they're also really safe to cook at high temperatures. They're less likely to be detrimental when you cook them at high temperature. So if you're cooking something at high temperature, a saturated fat is better. So like a butter or a lard or something along those lines. Unsaturated fats tend to be a little less stable, and they don't particularly like to be, they're not as safe if you heat them up at really high temperatures. So they're more of our low temperature cooking oils. With the exception, I should say, of macadamia nut oil, which is an unsaturated fat, but that one you can kind of heat up and it seems to be okay. The trans fats, these are the ones we want to stay away from. The trans fats have been partially saturated with hydrogen, and they do that in a laboratory. So this is not a naturally occurring fat, right? They add hydrogen to the fat to make it more shelf stable, if you will. And so these are the ones that seem to be the ones that are implicated in our negative lipid profiles. These are the ones that are associated with high cholesterol and, potentially atherosclerosis and heart disease and those kinds of things. So this is trans fat, right? So basically, like I said, they make these things in a lab, and they basically take the hydrogen and put it under a huge amount of pressure, and that is what causes those partially hydrogenated bonds, right? So basically what you're doing is you're taking a vegetable oil and you're making it a liquid fat into a solid fat. And this actually started a long time ago. You can see Crisco is the one that I think about, at least when I think about something you want to stay away from. It's a trans fat. This is what they fry things in, French fries or whatnot. If you ever work in a fast food place where you're frying a bunch of stuff, you'll see like these vats of trans fat. And the reason for that is because it stays stable. You can keep it in a vat for a long time, and it's, I guess, reasonably stable. I mean, I don't think it's good for us, but it's stable. It doesn't go bad. You can see Crisco was invented to make candles, and somehow along the line we decided to use it for cooking. I don't exactly know, but again, it's a kind of crazy process. What they basically do is they hydrogenate it, and what they do is they take the hydrogen gas and kind of push it through a catalyst, and that's what causes that liquid fat to turn into a solid fat. So we kind of consider these to be the ones that we want to stay away from, right? Because they lower the good cholesterol, which we call the high-density lipoprotein, and they raise the bad cholesterol. So even though fats are like kind of confusing, and lots of people are now pushing towards a higher fat, good fat diet, everyone agrees in the nutrition world that trans fat is definitely something we kind of want to stay away from. So no trans fats for us, right? Okay, switching gears, leaving the triglycerides and looking for a minute at the sterols and the waxes. So sterols, this is where we find our steroid hormones, cortisone being one of them, estrogen, testosterone, those guys. Progesterone is a steroid hormone. Pregnenolone is a steroid hormone. Vitamin D actually is a sterol. It's a steroid. It's a hormone like vitamin, which is interesting. And I think I mentioned cholesterol. So there's all kinds of things. We kind of traditionally, if we aren't that familiar, we think of cholesterol as being bad because we hear that a lot, but cholesterol is actually super important. It's a major communicator, right? Steroid hormones are our major communicators, and it's a really important component of our cell membranes. So it's not all bad. It just depends on where it is and how much you have. It's kind of a complicated topic. We'll talk a little bit about it later on, but just kind of keep in mind that this is a lipid. It's an energy molecule, but in this capacity, it's more of a communicator and a stabilizer. Waxes that don't so much have a role in the human body, but they do have a role in a lot of other animals, like this cutie right here, this little sea otter. Waxes kind of make this water repellent covering, right? So it keeps them from being too absorptive. We see waxes in plants as well. We'll talk a little bit about those later on. And that's a type of fat. Just again, it's a type of lipid. So here's a little more on cholesterol, right? We have kind of a working knowledge in terms of just news and such, health-related information that we hear enough about cholesterol, but oftentimes we don't really know. And the more we know, we learn that the picture is a lot more complicated. As a general rule of thumb, the good cholesterol is considered the HDL, or the high-density lipoprotein. It's mostly protein and not as much cholesterol. So in this picture, these green things are the proteins. These is the cell membrane here, and then the concentration of cholesterol is here. So it's not that much cholesterol, but it's got more protein. And you look at this one over here, the one that we consider not to be so good, the LDL cholesterol. And again, that's only part of the picture. There's different types of LDL cholesterol, some being worse than others. We're learning a lot more about this now. Sorry about that. Anyway, the LDL cholesterol is the one that we consider to be the bad one. Sorry, this phone is super annoying and I can't turn it off. Okay. Okay. Took care of that. So if you see here that essentially what happens is this cholesterol is moved to the liver, and the liver's job is to actually remove it. So, high cholesterol is in terms of the bad cholesterol, is sort of a tandem issue. It's got potentially a liver component and then this cholesterol component, right? But the LDL is the one that traditionally we think of as the bad cholesterol, because that's the one that's implicated even sort of loosely with atherosclerosis, which is plaquing of the arterial walls and ultimately leading to heart attacks. So we'll learn more about that when we talk about the cardiovascular system. Here's some simple recommendations. If you want to increase your HDL, that's the good one, and decrease the LDL, right? We want to eat saturated fats that will raise LDL. Those are things like avocados and good quality, butters and good quality fats like that. HDL is a really good one that we really want to get up high. That's where for sure, like olive oil is probably the best, shows to be the best in terms of raising HDL cholesterol. Carrots, nuts and seeds, like almonds, are good at raising HDL cholesterol. The omega-3 fatty acids, that's a whole nother story. Those are the good fatty acids that we find in the anti-inflammatory fatty acids that we find in fish, cold water fish specifically. And also we want to increase fiber, because fiber helps to remove the excess cholesterol, the excess fat. So foods that are high in fiber are whole grains, lots of vegetables, fibrous vegetables, and some fruits. So that's just a general rule of thumb. So fats can be kind of confusing because there's a lot of stuff happening in that category. Okay, then I want to really quickly talk to you about the proteins. Proteins are repeating units of amino acids. So like these green boxes would be our amino acid subunits that are going to repeat. So the amino acid subunits are going to be the same, and then there's like one thing that hangs off of each amino acid. We call it the R, as in rain, the R group. And what's happening at this configuration, this R configuration, is what makes the protein unique. Everything else is the same. All living things are made up of basically different configurations of 20 different amino acids. And the important thing, because the boxes are all the same. The thing that's different is the R group. So it's really 20 different R groups makes the proteins unique. So remember, proteins are, I said, the enzymes, they're structural, they make some hormones, there's a whole bunch of stuff that's happening. So again, this is the repeating amino acid subunit. You see it's exactly the same for all of these. This is glutamic acid, this is lysine, which is a different amino acid, this is serine, which is a different amino acid, but all this in the bluish box, they're all the same. This is the R group, so the R group is different. So it's the R group that makes the amino acid unique. And then we link these things together, kind of like linking a, like a beaded necklace, right? So you link the individual amino acids together. We're going to learn this. We're going to learn how to synthesize proteins in a few weeks, but it's going to be linking these things together through a very complex series of reactions. And then once it's linked together, the protein will start to maybe bend or coil in this case. And a lot of this has to do with hydrogen bonding. So this would be the most basic protein structure. They call it primary structure. That's like a pearl necklace. If it starts to kind of coil or bend like an accordion, they call that secondary structure. Still the same protein, just bent up. Then if it starts to get even more crazy in 3D, we call that tertiary structure. And then if you take different amino acids, or take different, protein molecules and link them together, like this one. So there's an orange one, this kind of yellowish one, this one, and this one. That's 4 different amino acids, or 4 different proteins, and they are linked together, and that would be called quaternary structure. That would be the most complex. So we'll look at those guys. So again, repeating units. Proteins interact a lot with one another. That's why they bond, that's why they fold, and they do all this crazy structural stuff. So a lot of the bonds that we've talked about are evidence on this picture here. So like, for example, lots of times when the protein will fold up like this, it's because of this attraction between this hydrogen hanging off here and the oxygen hanging off here, right? So that's a hydrogen bond here, right? This is another type of reaction that we find in things that are afraid of water, an hydrophobic situation. And those are called van der Waals interactions. If you read in your textbook, you read a little bit about those. This is a relation, kind of a bond between 2 sulfide groups. It's called a disulfide bridge, and this here is an ionic bond. So there's a lot of bonding that happens in proteins, right? And all of that bonding allows for that protein to have a pretty elaborate structural component, right? So you can see on this picture we've got hydrogen bonds, we've got ionic bonds, we've got van der Waals interactions, and we got these disulfide bridges, and all a disulfide bridge is essentially a covalent bond, but it's between 2 sulfur components, so that's why they call it a disulfide bridge. This is in between 2 cysteine amino acids, which are sort of sulfite amino acids, sulfur heavy. All right, so that's just, again, don't get hung up on all that detail. Just kind of understand that proteins are structural because of all these bonds. That's what I'm trying to tell you here. All right, when we have this protein folded up in a real unique way, we want to keep it that way. The shape of the protein is super important for the structure of the protein. If the protein's shape if the protein loses shape, it loses function. So if you recall when I was talking about pH in our class the other day, I said that if you change the pH of an environment, one of the things that happens is your enzymes stop working. So enzymes are proteins, and they're very sensitive to the pH. And so remember, we learned about pH last week, and all pH is a measure of how acidic or basic, or really what it's a measure of is the concentration of hydrogen bonds, or hydrogen ions, pardon me. And if you change the pH of an environment, it will be so detrimental because it causes the protein to unfold, essentially. And that process of unfolding or loss of structure is called denaturation. And essentially what you can, in your mind, think of denaturation equals loss of function, right? And they severely, when a protein is denatured, it severely challenges that particular protein, because essentially what it's doing is it's disrupting the relationship that our group is having to whatever it's attached to, right? So it's going to change, majorly change that shape of that protein. And the shape of the protein is super, super important. When we think of enzymes, we think of it kind of like a deadbolt in a lock. So if you jam some gum in there or something, that key's not going to get in, right? So that would severely impact the ability of that key to unlock that lock. And it's kind of the same thing. Protein's jobs, in this case, enzymes' jobs are to allow for reactions to occur. And if you mess up the enzyme, the reaction doesn't occur, right? So there's all kinds of things that will do it. pH and temperature are two really big factors, and also the change in the level of sodium that's around, or the salt that's around. And this, we'll learn that sodium is a very important ion that we find outside of all of our cells, and it's really important for the function of all of our cells. So by changing the pH, the sodium concentration, or the temperature of the environment, you can severely impact the ability of that enzyme to do its job. So enzymes are super important. We're going to learn a lot about them. They're basically these machines. They're always made up of proteins. That's what they are. They catalyze reactions, if you remember. They allow for reactions to occur. Enzymes are easy to identify because they all end in ASE. So example, this is lactase. This is the enzyme found in milk that breaks the milk sugar, which is lactose. This is a disaccharide, right? It's made up of two sugar molecules. One is galactose and the other is glucose. So the job of lactase is an enzyme that breaks this bond and it liberates these two monomers. So it basically digests sugar. All right, so most of our enzymes, almost all of them, I should say, are protein, but not all, but most enzymes are proteins, and we are going to learn a lot about them. All right, so here's just another thing in terms of what happens with temperature and meat and protein, right? So the question, why do we cook meat? Well, the obvious thing is cooking will kill things that are potentially infectious, like bacteria and pesticides, or parasites, pardon me. It also changes the flavor. So basically there's a reaction that happens when you cook meat. It's a chemical reaction between the individual amino acids. And basically what it does is it kind of reduces the sugar that are also found in those compounds, and it gives it a very yummy flavor. And that reaction is called a Maillard reaction. Right, so when anything that's browned essentially undergoes this kind of a reaction, and it's kind of interesting, right? But basically what it's doing is changing the shape, right? When you cook something, it's denatured, so you're changing the shape of those molecules, right? It doesn't always have to be high heating, right? You can cook things in a non-heat environment. You can do it in like when you make ceviche, and you put a whole bunch of lemon juice in there. Lemon juice is really acidic, and so basically what you're doing is you're basically cooking the meat with the lemon juice, right? If you've ever not made ceviche, what you do is you take a bunch of raw fish in most cases, and you throw it in this lemon or lime, lots of lemon or lime juice, and then you let it sit there, and basically the same kind of a thing happens. The proteins denature, and it's essentially cooking the meat, right? Salting meat, salted meat, all of that is the same, jerky, that kind of thing. Okay, so the very last thing I want to talk to you about are the nucleic acids. This is a picture of DNA, a DNA molecule. So the nucleic acids, if you remember, are RNA and DNA, and they are hugely important, probably the most important in terms of life of all the things that we've talked about so far. So they are made up of repeating units, like the carbohydrates and like excuse me, the proteins. There's two types, DNA and RNA. Deoxyribonucleic acid is DNA, and ribonucleic acid is RNA. They're made up of these nucleotide monomers, which is essentially, this is what a nucleotide looks like. I want you to remember this. Probably not for your first test, but for your second test, we'll remember this. So a nucleotide consists of, in the middle here, a 5-carbon sugar right there, and a phosphate group right here, and a nitrogenous base right here. There are only, in DNA and RNA, there are only 4 bases. In DNA and RNA, they are I meant to say 4 bases, pardon me, I think I said 5. In DNA and RNA, 3 of the 4 bases are always the same: cysteine, thymine, and adenine. And then we have one that's different, and that's uracil or guanine. So these guys are going to be repeating, and we'll learn more about this later on. They're going to be repeating as we go through the process of linking these individual nucleotides together. And the way that they line up is going to be hugely important. All right, so here you have DNA. Again, you can see the nucleotide, right? So basically what's going to happen is with DNA is a double-stranded molecule, meaning it's made up of 2 kind of pieces that gets linked in the middle, right? And the hydrogen bonds is what holds those 2 halves together. And when it links, it kind of coils up like a spiral staircase. The strands are complementary to one another, right? So they match, if you will. And we'll learn about how and why that is later on. It's not important for today. But what is important to note is the very bottom here, and the real significance of the DNA is that it has the entire genetic code. So I think I've said this already in our class, but every cell in the body has a complete set of DNA, has a complete set of blueprints, if you will, for everything that you need. And so that's why, you know, any cell that can be extracted, if you will, from you would have your entire genetic information, the complete set. We'll talk about why each cell kind of only works with a little bit of that genetic information, but every cell has it. So that's kind of an interesting thing. So we're going to get into DNA way more later on. Here's a little bit of RNA. 4 bases as well. RNA is a single-stranded molecule. It's really important because it allows for the DNA to be expressed, and we'll talk about that later on. The job of RNA is basically to get the genetic information and carry it to the place in the cell where it actually can be utilized. And that's the role of RNA. So we're going to talk about that. So for our purposes so far, all I want you to remember is that a nucleic acid is going to be either DNA or RNA, and that the repeating subunit is a nucleotide. So those are our 4 big macromolecules. We're going to get more of it as we go, but I just wanted to introduce them to you so when we do start talking about them in a lot more detail, they're familiar to you. So thanks for taking the time to get into this. Have a great holiday, and we will start talking about the cells when we get together next time. Thanks.